Welcome to the video on the DNA barcoding production pipeline, detailing the journey from sample collection to global sequence submission. The pipeline consists of four main phases: sample collection, DNA extraction and PCR, sequencing and barcode generation, and finally, comparison and database submission. A DNA barcode is a short, standardized region of genetic material used for global species identification. It functions like a retail barcode and establishes a universal language for cataloging biodiversity, reducing reliance on specialized identification methods. DNA barcoding has core applications in biodiversity monitoring, ensuring food safety by detecting mislabeled products, and assisting global law enforcement with forensic tracking of illegal trade. Phase 1 is sample collection. This crucial step involves acquiring biological tissue, recording essential environmental metadata, and securing the physical voucher specimen for verification. Field sampling requires precision. Every biological tissue sample must be accompanied by detailed metadata, including GPS, habitat descriptions, and preliminary taxonomic IDs. Accurate groundwork is vital for successful genomic analysis. Vouchering ensures a physical representative is deposited in a museum or herbarium, allowing sequences to be traced back to a verifiable source. Tissue stabilization immediately halts DNA degradation using methods like 95% ethanol or flash freezing. In the lab, preserved samples undergo chemical DNA extraction. Cellular membranes are lysed, proteins are removed, and purified DNA is precipitated, creating the genetic template needed for accurate amplification. DNA barcoding targets a small, standardized fragment, the 658 base pair region of the mitochondrial COI gene for most animals. This universal fragment is highly conserved. Phase 2 focuses on sequence amplification. This involves isolating and exponentially copying the specific barcode region using the polymerase chain reaction, or PCR. PCR involves denaturation to separate DNA strands, annealing where primers bind, and extension where Taq polymerase synthesizes new strands. Repeating this cycle 30 to 40 times achieves exponential growth, yielding millions of barcode copies. Different taxonomic kingdoms use specific marker genes for barcoding. Animals use the COI gene, plants use RBCL and MATK genes, and fungi utilize the ITS region. Before sequencing, PCR success is verified by electrophoresis. The amplified DNA fragments are separated by size in an agarose gel under electric current. A distinct fluorescent band confirms the presence of the targeted approximately 650 BP barcode. Sequencing technologies vary. Sanger is the gold standard for precision individual barcodes. Minion offers real-time and portable sequencing, and Illumina NGS allows for massive throughput, enabling meta-barcoding of thousands of samples simultaneously. Phase 3 is the generation of the barcode. This phase translates chemical sequences into digital data and executes complex bioinformatics workflows to finalize the barcode sequence. Bioinformatics processing begins with trace editing, where specialized software trims low-quality data from raw sequences. Next, contig assembly aligns and merges forward and reverse reads to generate a highly accurate consensus sequence, the final digital barcode. Analyzing genetic divergence requires calculating the evolutionary distance between sequences. The Kimura-2 parameter, or K2P model, is a standard metric used in barcoding that accounts for different mutational rates in this process. The quality assurance workflow involves translation to check for pseudogenes, alignment against standard models, a blast check for contamination, and final validation before global submission. Phase 4 is comparison and submission. This is where validated data contributes to global repositories and helps identify unknown specimens, completing the entire process. Validated data is submitted to global reference libraries. Bold Systems is strictly for barcoding and requires interconnected data. GenBank is a comprehensive public database where sequences are synchronized internationally. The vast majority of global barcode submissions currently belong to arthropods. This reflects worldwide initiatives to catalog highly diverse and severely understudied invertebrate populations.